Hello friends, welcome to the second class of replication, and in the first class we have discussed about the replication origin, and there we talked about the 9 base pair long repeat sequences and 13 base pair long repeat sequences, and the DNA double helix and semi-conservative, how this DNA double helix are holded by the double-stranded uh, in this double-stranded structure by the hydrogen bonding and then DNA melting and the conservative and semi-conservative replication. And there we told about the helicase protein, but I made a mistake over here that this is not DNA A protein. The helicase function is shown by the protein called DNA B. So now what is DNA A? DNA A, which binds into this 9 base pair repeat sequence. DNA A protein is basically the works as a single stranded binding protein. So we have to discuss on about that, and this is the place where we can draw some DNA. And so this is the double-stranded helix of the DNA, and which is highly coiled structure. So when the helicase, which is DNA B, breaks this hydrogen bonds over here, this is the hydrogen bonds, and when it is broken, it forms a replication bubble. And suppose this is my fork, and here the bond has been broken. These are the remain this is the place, this is the single stranded structure, this is the double bonded structure still here, and here is the protein which is called helicase, which is moving in this direction and opening, unzipping the DNA structure, which is the DNA B protein, uh, and in this single stranded DNA we'll be able to find out some proteins which are associated with the single strand, and these proteins are called the single stranded binding protein, or we can write it as SSBP in small. And this is basically the DNA A protein. Now why it is necessary? Because as it was in a very much coiled form, so when this double bond, this double stranded structure breaks, this single strands of the DNA has a tendency to recoil in their own structure, and if it happens it will hamper the replication process. So to hold these strands straight in the single stranded form, these proteins get associated with these single strands. Here also there are some SSBP protein hanging with this strand. It is keeping this strand straight so that the replication process takes place smoothly, and DNA A proteins are the proteins that remain associated with this 9 base pair long repeat sequences. And there it keeps the when the DNA B works and the DNA breaks into the single stranded form and makes the replication fork, then this DNA or SSBP hangs with the single stranded structure to keep it single and simple. So now the thing is that this all replication process that we are discussing so far, it is in the E. coli, which is a prokaryote. And what happens, well, in the start of the DNA, this is the DNA strand has been already opened, and now a small RNA. Okay, let me clear this portion. Okay, now what happens? A small RNA primer is formed over here. If this is the 5, this is the 3 prime structure, this is the 5 prime end, and then this is again the 5 prime end, and this is the 3 prime. One thing I should say, that this replication process takes place from 5 prime to 3 prime direction. Okay, so in this strand where the 3 prime end is here, so this portion here, the new RNA, small fragment of RNA, which can hang with this hang with this DNA strand is 5 prime to 3 prime. This is the orientation of the new RNA. Why? Because the a DNA can only tolerate a structure which is anti-parallel. That means if this is the 3 prime and the end that will be with this 3 prime end has to be 5 prime. So this new synthesized RNA has to place its 5 prime end to this portion and 3 prime end to this portion, and the DNA synthesis will take place in this direction, as we have already discussed that this process takes place from 5 prime to 3 prime direction. Now, why this RNA is here? RNA works as a template. Okay, the DNA polymerase that polymerizes the nucleotide, taking this other strand of the DNA as a template. It cannot start its replication process on its own. It needs RNA as a template. So this is the RNA, the DNA polymerase that actually replicates the DNA is a RNA dependent DNA polymerase, and here is the RNA primer, and this RNA primer is synthesized by the enzyme called Primase. Okay, this is Primase that synthesizes this RNA template, and in E. coli it is the DNA G, okay, which is called as a primase, and it synthesizes the RNA template. Now there are three kinds of DNA polymerase exist. Now one is, okay, let me take it into a new whiteboard. The first one is the, okay, this is the name and this is the function. All right, I'm talking about this first DNA. Please bear with me with my handwriting. DNA pol 1. This is the DNA pol 1, and it functions as the as you know, filling up the uh, the small portion of the DNA segment. Okay, so this is not a major DNA pol enzyme. It actually fills up small portion of the DNA, or it can functions as the DNA repair enzyme. So the main function is DNA repair and filling up, repair and filling, and filling gaps. Okay, now the next is the pol 2 enzyme, which is mainly associated with the function that is DNA repair, and the third one is the most important one because it is completely devoted for the replication, of both leading strand and lagging strand. Now here comes the next question: what is leading strand and what is lagging strand? Now if you look at this one, suppose we have already, you know, put our okay, let me take a new color. So let me take this one. So this is my DNA polymerase 3. Over here I have put this enzyme over here, and it will move from this 5 prime to 3 prime end, and the road is quite clear because we have already got a 3 prime template on our side. So when the replication takes place, it will continuously synthesize this strand without any problem, and so this kind of strand which can be synthesized continuously is called a leading strand. This is called the leading strand. Okay, leading, I guess you can understand. So this is a leading strand. On the other hand, if we look at this structure, this is the 5 prime end and this is the 3 prime end over here, and we cannot put sorry, let me increase the timing of this class. If we have to put this RNA structure over here somewhere, it will be 3 prime and 5 prime, and and DNA replication cannot take place in this direction. It is not possible. And if it takes only in 5 prime to 3 prime direction, so in this direction it cannot be possible either, because there is no template. So what happens to to solve this problem? What happens that this template is placed somewhere here, uh, and then again somewhere here. So if this is the 5 prime end and this is the 3 prime end, and again if this is the 5 prime end and this is the 3 prime end, then the DNA can be this DNA. Okay, let me take this color. Now this will be the same color, so it's better that I would take this red only. And so the new strand of DNA will be synthesized from this 5 prime to 3 prime up to the uh, RNA segment, and then again this 5 prime to 3 prime up to the end. So there are small fragments, so that strand which cannot take place in the continuous manner, again it has to wait for this helicase. When it will again move to this direction, it will again get some place to place another RNA template and to synthesize another fragment of the DNA. So in this case, the strand synthesis doesn't take place continuously, it is in a fragmented form, and this strand is called a lagging strand. Okay, this is the lagging strand and this is the leading strand, and this lagging strand, these small fragments are called the Okazaki fragment. This is the Okazaki fragment, and this whole synthesis of the leading strand and lagging strand is done under the control of the DNA polymerase. Okay, so if we go back to that slide, here is the DNA pol 1. We have known that the DNA repair and filling of the gaps is the function of this DNA pol 1, DNA pol 2, DNA pol 3, and there are two more DNA pol 4 and 5 exists, which have no proofreading ability and replicate the damaged DNA and belong to the gamma family. So they actually are responsible for the replication of the damaged DNA. So damaged DNA replication, because they lack the proofreading activity. And now what is this proofreading activity and everything I will be discussing in my next video, so stay tuned. Thank you.